So my presentation will be divided in 3 sections. The first section, I will give overview about the disease and the main strategy to manage this disease. And then I'll follow with strategies that breeding has used to manage this disease. And I'll finish showing some of the work that we are doing towards a new approach and towards the understanding of the pathogen. Okay, so that's the idea. Just to introduce you, to give idea how important this is just a picture that I took at a Ponta Grossa, Paraná State in a chemical field test station. You can see here there's a lot of plots in which they are testing different fungicide. We have here a plot without chemical control and a plot of chemical control. You can see the difference. Phacops opaciae, that is the pathogen that causes the Asian soybean rust, mainly attacks the leaves, causing this defoliation, very strong defoliation, you can see here. So you can see that plant will not be able to do the photosynthesis process at the level that it would be able to do, so you have a decrease in the production, okay? So chemical control, we're going to see, is the main strategy to control this disease in Brazil. The disease is present, the pathogen is present in all the soybean growing regions in Brazil. You can see here the Cerrado region, and then you have the south part of Brazil. You can see the disease is always present every year. But depending on the climate, the disease can be worse or less worse, okay? So you can see the disease pressure normally is high in the south part of Brazil because we have this disease problem in our neighbors. For example, Paraguay and, for example, Bolivia. So sometimes you have a inoculum pressure from this place that also contributes to the increase of the import of the disease. I forgot to explain to you this information I got from the Anti-Rust Consortium page. And this red color means that at that place was reported soybean rust, okay? At the beginning of the season. It's just to give idea to the farmers if the disease is present and they can use this information to decide if they are going to start to spray or not to spray, okay? Just to give idea. So overall, the disease is present in all the areas and has a very strong potential to decrease the productivity. So the farmers has normally they control using different strategies. Just to give you a feedback in plant pathogen, the amount of disease that we see in a field or in a region depends on the pathogen, how abundant is the pathogen, how virulent is the pathogen, depends on the environment. Especially for Asian soybean rust, humidity is very important, and the temperature is 25 to 27 degrees is the best condition, and also the susceptibility of the host and how abundant is the host. Soybean rust Phacops do not have a problem of host in Brazil because you grow soybean in more than 33 million of hectares, okay? And then we have our neighbors that also grow soybean. And we have also some hosts like perennial soybean. That is a very good host for soybean, okay? So when all these 3 factors interact in an optimal condition, we have a condition to have an epidemic, explosion of the disease in the field, okay? So all the management is direct to any one of these 3 components direct to the pathogen, direct to the environment, or direct to the host. We have here in Brazil a very important management strategy. It's a legislative strategy. We cannot grow soybean or to have soybean for 6 to 90 days after soybean crop to decrease the amount of pathogen that we have in the field. Okay, so it's a very important. In many places in Brazil, the government, state government, ensure that the farmers follow this legislation. And then we have immunization, the principal immunization that we're going to talk a little bit, that is based on genetic control. And then we have the chemical control, is in the wrong place here. We have the host that's susceptible. We can protect the host using chemical. And then a very important strategy is avoidance. In Portuguese, evasão ou escape. It's a very important strategy nowadays. By using short cycle varieties, we can escape the environment conditions that are favorable to disease. And in some place, the farmers can go to the end of the cycle without any spray. Okay, I'm going to talk a little bit because the purpose of breeding short cycle soybean was not to control soybean, but nowadays a very important strategy to control. So we can discuss many issues here. There's a lot of issues related to each control strategy that we are using. But I'll leave here just to the end. We can discuss a little bit about that. We can discuss why resistant variety is not commonly used in Brazil. Why it takes so long to have a new fungicide registered here in Brazil compared to the other one, and many other important issues. So just to give you an overview of the importance, we spend about $2.4 billion in fungicide here in Brazil to control just Asian soybean rust. Why this amount of money going to control just one disease? Because you grow soybean in 34 million of hectares in Brazil. I can imagine one spray. Normally, in average, 4 sprays during the season is used to control soybean, rust, and other disease. So this large use of fungicide has a price because we spray a lot. We expose the pathogen population to this amount of different chemical, and this procedure can select strains that are not very sensitive to the fungicide. What we are seeing year after year is an increase in the resistance of the Phacopsera to fungicide. You can see here a program that was used in 2006-2010 nowadays cannot provide a very effective control of this disease. And you can see now a program 2011-2015 is not doing so well also compared to the other program here. Why is this happening? Because of fungicide resistance. What we are seeing is a decrease in the efficiency of control of the disease because the pathogen is able to develop resistance to the major single-site fungicide that we are using here in Brazil. So because of that, other control strategies are becoming more and more important. One very important strategy, as I said before, is to use short cycle varieties. You can see here, this is the disease development around in different growing season. You can see the disease start to be important in December, and the peak is February. If you plant a very short cycle variety, you can escape this stage here. Just to give an example, If we plant a short cycle variety in September here, 10th of September, now we can have the growth development and the cycle finish before the disease become important. If you use a different date with the same variety, different plant date with a different variety, you can see that we are moving inside of the storm. If you go later, you are going to have a problem. You have to use a lot of fungicide to spray. So that's a strategy that is used today, but the purpose, as I said, was not to control soybean. The main purpose was to allow a second soybean crop. In many of the places, the farmers grow corn nowadays or cotton after soybean. So if they plant soybean early, September, short cycle soybean, they can harvest soybean beginning of January, and after this plant, they can plant corn or plant cotton and have a second crop. So that's an important strategy. But this strategy used for this purpose is giving a very huge impact in the soybean control, especially in the Cerrado region, Mato Grosso and Goiás. I'm giving this example here because you can see sometimes we plan to control one disease developed disease-resistant variety, but you can see that you can control a disease sometimes work with the variety cycle or variety architecture and other steps that are very, very important for disease development. So the other strategy that I'm going to go in detail, I'm trying to go a little bit faster, is disease resistance. But compared to abiotic stress, especially here biotic stress in my particular case, disease resistant, the phenotype is not dependent on the plant genotype and the environment only, okay? We have now another component here, that is the pathogen genotype. That makes abiotic stress a little bit challenging compared to abiotic stress, but I know the abiotic stress is a very complex trait to work. But just to understand, when we work with disease resistance, we have to think about the plant genotype and also the pathogen genotype. Because the same genotype in the same environment, if we change the pathogen genotype, we can see changes in the phenotype. That's very important for any purpose, to study variability of the pathogen and also to map genes, because if we change the isolate during our mapping work, we are going to see results different. So I'm going to talk a little bit about the plant, and then I'll finish some of the work that we are doing for the plant, trying to understand the contribution of the pathogen for the disease. So another big point very important point for disease resistance, that we have to understand that disease resistance is not everything, okay? The farm is not to grow a resistant variety just because this variety is resistant. This variety has to be very productive and it has to be very reliable in year with disease and year without disease. Because if the variety is not very productive, they are going to use a susceptible variety and they are going to use a fungicide. And at the end, the result will be better compared to the result that he could get plant a resistant variety, but it's not a productive variety. Another big issue in plant pathology, especially for breeding for disease resistance, that we are looking for durable disease resistance. That disease that is is stable for during the long use of the variety and the use in the variety that the environment is favorable to disease. Okay, it's like a dream. We can talk a little bit about that later. So in soybean, there is two different strategies to control. I'm going to talk a little bit about three strategies that can be used to control Asian soybean rust by using genetic strategies. One is to use native genes. In soybean, you have this is already described 7 loci called RPP1 to RPP7. We have dominant recessive gene, resistant gene is this loci, just to give overview of these different loci. These resistant genes that are located in these loci can provide different phenotypes of resistance. And also many of them just provide resistance that is isolate specific. So many of genes are already available that can be used for traditional breeding and introgress in commercial varieties. Okay, just to give overview, the phenotype, this is the susceptible phenotype that is called tan color, because of this type of color. You have the immune phenotype, when the inoculate plants do not show any symptoms, and you have this RB hedge brown phenotype, in which the inoculate plants show these head lesions here at the penetration point. It's like a hypersensitive response. In some of the genotype, you can see some sporulation, some we don't see sporulation. Those are the main phenotype. This resistant gene has been used, but we don't have many resistant varieties in the market. One of the main reasons that is most of the material at the beginning did not yield very well compared to the susceptible varieties. One reason, I believe, was linkage drag, because most of the source of resistance are not adapted to the Brazilian condition. They are from China, Japan, and other places. So they are more adapted to the environmental condition from those places. So when we introgress the gene, sometimes we introgress together allelic form of genes that are linked to the resistant loci that can provide some undesirable traits. Among of them, low yield. So sometimes you have a resistant variety, but the variety is still not very productive compared to the susceptible variety. So what we are doing in the lab is collaborate with some companies. We are using molecular markers to narrow or to reduce this linkage drag, or to develop markers that during the selection, the linkage drag that can be very small, and then the variety can be productive compared to the susceptible one. That's very important, because another strategy to breed for disease resistance, to have a more stable resistance, to combine different resistant genes in the same variety. You can see if you combine different genes, the problem of linkage drag will be worse. Okay, so this approach has been successful. Just to give overview here, there's a company called Tropical Melhoramento Genético that has been breeding for Asian soybean rust resistance. You can see here in 2018 we had a very high pressure for Asian soybean rust. This is a field with a susceptible variety, and this is a field with a resistant variety. You can see that both fields were sprayed. The difference here is because of the rain, they could not spray this field at the right time. Okay, they lost the right time of control here. In this case, the resistant holds the disease until the weather condition was allowed the sprayers to be used in the field. Okay, so that's important. Sometimes it's not important to have the control, but have some insurance that we can slow down the progress of disease for sometimes that then we can use a different strategy. Okay, so Embrap is also developed some resistant varieties. A tradition is not a transgenic resistant variety. You can see it's susceptible, and then you have here resistant variety that has this RB type of reaction. Both are inoculated. You can see here, you can see sporulation. Here you don't have sporulation. Okay, but the problem with this resistance based on native trait, that disease can be overcome by new race of Phacops. Just to give idea, this variety from Embrapa has the RPP5 gene, and this isolate here can overcome the RPP5 gene. Okay, we can find in the field, even in place that this variety was not used, isolate of this pathogen that is able to overcome the resistance. So another strategy that we are using is to look for new resistant gene from tropical legumes. Phacops pachyhyza has a very broad host range. There is more than 156 species that are described as a host of Phacops. You can see many of them here. We have a Cajanus cajan, or pigeon pea. We have a Vignia, cow pea. We have a Phaseolus, lotus. All these plants are reported to be susceptible, but just a warning, be careful what people report to be susceptible, because sometimes you cannot repeat the experiments, okay? So the idea behind of this approach was to find genes in closely related species and use transformation to transfer this gene from one species to the other one. Okay, so the idea was to look for genes that can provide broad spectrum resistance, or gene that has some new mode of action and trans to soybean in order to get a very strong resistance. So that's some of the work that we are doing. For example, in Cajanus, we look inside of the genetic variability of Cajanus and look for plants that were susceptible or resistant to Phacops, to our standard isolate, because we need different phenotype to do crosses to understand how many genes are involved in the control of the phenotype, and also to map the gene and clone the gene. If the resistance is polygenic, it's very difficult to transfer by transformation to cultivate soybean. Okay, so we were able to find some immune genotype, some very susceptible genotype. By crossing these two genotypes, we were able to find a genotype of single disease-resistant gene. So we use, nowadays we can use that we call next generation genetics, using all this new sequencing technology to develop markers and to map the genes, to find genes that are very close to your target gene, and use this information to identify and test candidate genes. And then you can transform soybean, okay? So the assumption behind this approach, the gene has to work in soybean, because if it works in Cajan but do not work in soybean, it's not useful. Has to provide very broad spectrum resistance, because if you do not provide very broad spectrum resistance, why are you going to spend money transfer this gene to soybean? You go to all the regulation process that are very expensive. So ideally, the gene has to have a different mechanism compared to the genes that we have in soybean. So this strategy, we have a proof of concept. It works. We're able to identify and clone a disease-resistant gene from Cajanus cajan, transfer this gene to soybean, and this gene in soybean was able to provide the same level of resistance that we see in Cajanus cajan. And another interesting issue about this gene is this gene was able to provide resistance to a broad number of isolates that we had collected in many different growing regions here in Brazil. Okay, this work was a collaboration with Corteva and Two Blades Foundation. Now we thanks a lot to Corteva to believe in our work at the beginning and to give us support for this development. We are continuing to look in other species, another gene in another species like related to soybean, and approach seems to work every time. Okay. Another strategy that people are thinking about to use for disease control is gene editing. I put here cell smoke because at the moment we do not have very many examples of using this strategy for disease control, at least for fungal disease. So far, this gene targeting is a limiting factor when you try to replace a susceptible allele by a resistant allele. So far, it works only for genes knockouts. It means that we can use this technique to knock out some gene. If the gene is important for Phacops infection, by mutate this gene, we can get some resistance. So we are looking for targets. How we are looking for targets? We are doing a very broad mutagenesis study. We have mutagenized a susceptible soybean variety or genotype. We use ethyl methylsulfonate. And we are screening this population with specific isolate, looking for a gain of resistance. Just to give idea, this progeny from one T0 plant that we have inoculated, you can see here a different kind of reaction. So it's the kind of reaction that we are looking for. And then we have to go after the gene, try to know what's the function of the gene, or if this mutation, this gene can have some impact in other functions that are important for the plant development. We have found very interesting phenotype. Okay, you can see here that we have some mutant that we have this nice target lesion. You can see this plant has been inoculated, and we have this line of defense around the infection point. Or sometimes we have this yellow color around the infection point. And normally in both cases, we can see a very low level of sporulation. Okay? You can understand that this mutation here, in the way that I'm showing you, is not useful for breeding. But to understand the interaction is very important. To understand gene that is important for interaction is very important. We can use different approach. Again, use this new sequence technology. It's possible in a very short time to identify the specific gene that was mutated. Okay, we are working with 4 different genes in these 4 different genes. We were able to find the candidate gene for the trait. So Another line of work that we have in the lab is work aimed to understand how this RPP gene works and how Phacopsia pachyhyza is able to overcome them. Just brief overview. You can see during the infection, we know that the pathogen are able to secrete and translocate that we call effectors. If the plant has the resistant gene, some of these effectors are recognized by the resistant gene, then you have the resistant phenotype. But the primary purpose of this effect is to manipulate the host, to avoid that the host respond with resistance, or to make the host make nutrients more available for the pathogen development. If that occurs, then we have disease. We call this gene that are manipulated by the pathogen, we call this as susceptibility genes. As I said before, genes that we are looking for, that we could mutate this gene in order to get resistance, because the pathogen could not manipulate this. Just to give an example of effector of Phacopsia, this effector is targeted to the nucleus. Just show here is fusion with green fluorescent protein. This is a protein that we know that is targeted to the nucleus. And then we combine, you see both colocalized indicate this protein is targeted really, is targeted to the nucleus, okay? This gene is expressed just 24 hours after inoculation, after the pathogen starts to ingress in the pathogen. To help to understand more the biology of Phacopsia, there is a consortium that is aiming, was able to sequence 3 different isolates of Phacopsia. So the genome is available. We have a sequence full reference of a new 2 isolates, and we have a sequence at least 40 different isolates. Okay, so improvement in the technology in the last 5 or 10 years is allowed to do something that people was not able to do before. There was an attempt to sequence Phacopsia pachyhyza few years ago by JGI, was not successful because the technology that was available at that time. So that's the consortium. Just to give overview, Phacopsia has a very large genome. Compared to Magna Porti, it's just small here. The genome size of Phacopsia is almost the genome size of soybean, around 1.1, 1.2 gigabase of DNA. So Phacopsia has dikaryotic spores, so you can see a lot of heterozygosity. So it's like a hind variability that Phacopsia can be used. Just to give you an overview here, you can see the heterozygosity. Just by looking at the SNP, you have 50% of red and 50% blue, indicate that we have 2 different alleles at this specific position. So we have a collection of different isolates that we now can profit of this reference genome. We can now look for genes or look for changes that are involved in the process of overcoming the resistance provided by RPP genes. And also to understand other mechanisms that Phacopsia can use to overcome disease resistance. That is, although we don't have a sexual reproduction in Brazil, there is some mechanism that Phacopsia can use to recombine or to bring together different virulence genes that are present in different isolates. Okay, so I'm going to finish here because I over my time, and as I said, I will leave a question sometimes for question now if I have time, or you can answer any question during the section. Thank you very much for your attention.
Thank you, Professor Brommonschenkel, for your nice talk. It's really interesting, this pathosystem. And we have some questions here for you already. James Bori is asking what genomic approaches you have used to map the resistant gene in pigeon pea.
Okay, just let me finish here. I am going to turn. Okay. Basically, what we are doing is to sequence the parent using these new technologies. Nowadays, PacBio is the way to go because we have a hi-fi, but at that time we did not use hi-fi technology, and we use just reference genome as other species reference, and we map the breeds from Cajanos. But nowadays, the way to go is PacBio, hi-fi, both parents, compare their parents, look for SNPs, and use these SNPs as markers to map the gene. Do a whole genome scanning for linkage to specific resistant gene. So that's the approach now to go. Because it's so I think the big change now, as I said, next generation genomics is to be able to do in a very short time that in the past we used to spend years to develop genetic maps using different kind of markers. Michel Moore here, he spent a lot of time developing maps, lettuce map using R-Philippines and other different types of markers. Nowadays, with sequence, it's getting much easier to do mapping for any traits. If you, the major point is phenotype. You have to do a very good phenotype, because if you don't do a very good phenotype, your gene is going to be moving to one place to the other. Okay.
Okay. Thank you. I have one question for you. Well, I have a lot of questions for you, but of course I cannot do all of them right now. I would like you to talk a little bit more about the variability, the pathogen variability. For us in plant breeding, this is a major concern. And it's a huge task, it's a huge challenge. And I would like you to talk a little bit more about it. There is a program to monitoring prevalent races around Brazil.
Yes, we have in the lab every year, we collect isolates in many different places in Brazil, and we test them on the genotypes that have a known resistant gene. But also this survey is not very precise because the population of this pattern is so high in the field. Okay, so doing the same way that we always see now at TV about COVID-19, that Brazil is a very good place to get new strains from COVID-19. Phacops is a similar situation. We have a very broad number of hosts in Brazil, soybean, 34 million of hectares. So the inoculation is very efficient. You can see if you have a spore in the air, if the spore lands, there is a very high chance of landing in a soybean plant, especially in those areas. Okay, so in my opinion, mutation is the main mechanism that Phacops are used to adapt to fungicides or resistant variety. And you can see mutation is not derived by selection. It's a process that normally occurs. And the higher the population, the higher the frequency of mutation. So the key point to control soybean is to keep the population size very small. Okay? So that's host-free period is very important. That's why fungicide is very important. That's why resistant varieties are very important. We can protect the resistant variety using fungicide, and the resistant can protect the fungicide, because both will decrease the population size of the pathogen. So that's the idea. To have a stable Asian soybean rust control, we have to use both fungicide and resistance. If you just use one, we are going to fail. As we have failed for monogenic disease resistance and single-site fungicide.
Yes, resistant cultivar is just only one step to get plants healthy. So it's part of an integrated management, and it's important not to expose all resistant genes at one time. Otherwise, you get the genes useless for breeding.
Yes.
In a short amount of time.
Especially in our system that we have a very genetic uniformity. Now you can see we grow millions of hectares of the same variety. You can imagine if you put millions of hectares of one resistant variety with a single resistant gene. So just one season, the resistance will be broke. Okay,
we have another questions. We have other questions here. Rogério Marges is saying, nice presentation, Sergio. I would like to know if Pachypsora can also infect neotropical legume trees like Schizolobium or others.
So, Rogério, thank you. It's a very good question. We have not tested yet, okay? But there is some trees that are reported, especially legume trees that are reported to be susceptible. But as I said, just be careful. What people say that's susceptible, sometimes just lesion. In my opinion, we have to be able to find sporulation, enough sporulation, because we normally call a plant susceptible is a plant when the pathogen can complete the whole cycle, okay? But it's possible, it's possible. Well, I didn't have time to look at that plant because, but there is some report in the literature that can be infected.
Okay. Professor Silvaldo is asking about if there is a reason for the occurrence of sexual phase of the pathogen here in Brazil, and if there is some research that compares the variability in Brazil, if it's equal or where the sexual phase also occurs.
Okay. Thank you for your question, Silvaldo. It's a pleasure to have you attending this symposium. Silvaldo was my first student. I'm not sold, but he was very good. Actually, in Brazil, we can find teliospores. Okay, you can find teliospore. I do not know anybody that was able to germinate those teliospores. In other places, also, like in Japan, was reported to be in some place, in some infect plant, you can find teliospores, okay? But no one was able to infect soybean with teliospores, okay, with basidiospores. It means that possibly there is another host, in which soybean Phacops can complete the cycle, okay? But has not been found yet, okay? Regarding the variability, the population of soybean in Brazil, Phacops is a clonal population, but as I said, mutation is occurring all the time. You can see noise of mutation in different isolate. But if you look at the isolate as a whole, basically is a clonal population. Okay, but there's a lot of variability. Even being clonal, you can find isolate that can overcome a specific resistant gene. Okay. Remember that I said also the pattern is very heterozygous. So you can keep a virulence gene in a heterozygote condition. You can express this gene when it's necessary. Okay, it's something that we are looking for now. Okay, but basically it's clonal but variable. Don't think that clonal means equal. It means that the level of variation is not so high, but still high enough to cause trouble, like a fungicide resistant or overcome resistant genes.
Okay, thank you very much, Professor Brommonschenkel.